Plants die when you don't water them. Gardeners and farmers know this, and in a world of climate change and ever-increasing drought, this is becoming a more critical issue. However, a small number of plants, scattered across the plant kingdom and across the world, have managed to solve the problem of water shortage. These so-called resurrection plants can survive losing almost all their water and recover full physiological activity afterwards. This beautiful Xerophyta schlechteri is an example. Here you can see it recovering from drying after I've watered it on this very table up here in my attic. As you can see, resurrection plants do not function like succulents, which are adapted to retain water in dry environments. Instead, resurrection plants happily surrender water and, just like seeds, enter a state of dry, suspended animation, the state we call anhydrobiosis. Now, as I'm sure you know, water is fundamental to life on Earth. To survive without it, for any plant or animal or fungus or bacterium to live without water, calls for many adaptations on the levels of anatomy, physiology, and genetics. This whole question of anhydrobiosis has kept many biologists busy for years, including many wonderful colleagues, collaborators, and mentors of mine. But my particular little corner of this puzzle concerns the way in which small molecules, like sugars, amino acids, and other simple metabolites, help to keep the plant cells stable when they're dry. And what I've done is to measure the metabolite profiles as they dry down of a whole wide variety of different species of resurrection plant, ranging from mosses to complex flowering plants like these. So here's what we found, and here's the questions that it raises next. First of all, as you can see in the PCA plot that I'm showing here, so-called normal metabolism is very diverse, but resurrection plants in the dry state are very much less variable. That's to say, as the plants go through their days under normal physiological conditions with enough water, dealing with varying light, moisture, energy status, temperature, and so forth, their metabolic profiles can move around quite a lot. But once resurrection plants start losing a large proportion of their water, they converge onto a much more narrowly defined set of conditions in terms of their metabolite profiles. When we look closer, we see that there are really two strategies involved. One based around accumulating a large amount of the unusual sugar trehalose. This is the same sugar that is accumulated in desiccation-tolerant fungi and those few animals that can also do that trick. This seems to be the more ancient and ancestral of the two strategies. The other strategy that we mostly find in flowering plants involves plants accumulating massive amounts of sucrose supplemented by higher molecular weight sugars like raffinose and stachyose. In this group, it is these so-called raffinose family oligosaccharides that seem to make the difference between life and death. Indeed, the amount of raffinose is the only major metabolic difference between a resurrection plant like Xerophyta here and the desiccation-sensitive Arabidopsis thaliana. So, sugars are not the only small metabolites that respond to water loss in resurrection plants. We also see important differences in the profiles of amino acids, organic acids like malic and citric acid, and various antioxidants, chlorophyll breakdown products, and micropeptides. The question now is, given how much these plants vary, how many distinctive features each species has, anatomically, genetically, and biochemically, why and how do they all seem to converge reliably every time on these narrowly defined states? To start answering that question, I'm taking two approaches. The first is to use the metabolite profiling data that I've generated to model changes in metabolic pathway activation and to test these predictive models against both gene expression and enzyme activities in as many of these species as I can reliably get data for. The second approach is to look at the physical characteristics of artificial mixtures of these important molecular species at the same molar ratios that we find in cells. We know that the thermodynamic properties of cytoplasm in anhydrobiotic states are very distinctive, with cells forming either a stable glass or else non-aqueous ionic liquids that may fill in some of the functions of the missing water. Will it work? Well, I'll tell you next year. One final question, of course, is why we put so much time and energy into these studying these fairly obscure plants. And it's true that the world is facing more and more frequent and more severe droughts. So everything that we can learn about plant stress tolerance and water deficit stress in particular could potentially improve crop improvement programs. But the real reason I study them is because they're beautiful. There is something inspiring about these wonderful organisms that hunker down and survive the worst that nature can throw at them, then simply pull themselves back together and carry on when favorable conditions return. After all, we scientists are people, and hopefully we do what we do because we intensely and deeply love the world we're in. Thank you for your time. Please feel free to get in touch if you find this work interesting, and hopefully we'll actually get to see each other next time around.